All right, so in this video, we're going to spend some time talking about this, which is the genetic code. This is one of the fundamental parts of biology. So, and I can see that my picture is blocking part of it. So hold on, let me move myself out of the way. If I can here, hold on. There we go. All right, so, all right, what is this? So this is a table of all the possible three-letter words you can get from the letters G, A, C, and U. So if you take those four letters and write them in a list of every possible way of arranging them, U, U, A, U, G, U, C, U, C, U, U, C, A, U, C, every possible permutation you can get, there's actually 64 ways you can write those letters as three-letter words. They don't mean anything in English, but there's still 64 different combinations. And they're all listed out here. We could list them as a long list of 64, but it would be very long and it would be hard to scan through it. It's kind of easier to display it like a table, like you see it in front of you here. So basically, if you look at how it's written, it's using U's. So clearly this is, clearly this is RNA that we're looking at, right? So this is a table of codons. And if you remember, this is really important to remember from the video about translation, the codons are the three-letter words on the messenger RNA strand, that pink strand that was running through the ribosome. We started off reading it with the first AUG, which we called the start codon. And then we put groups of three at a time through the ribosome. Those groups of three at a time, nucleotides, are called codons. And they're always found on the mRNA. All right, so that's the first thing to understand. Now, if we look at this table, you can see the three-letter words, U, C, UUA, UG. These are our codons in the capital letters, right? And then there's these brackets over here, grouping them together with these other words. Now these three-letter words are amino acid names. So this is like a translation, like a dictionary that helps you translate between one language and the other. This is the language of nucleotides. This is the language of amino acids. You do not have to memorize this. You don't even have to memorize these abbreviations. Anytime you'll need to use it, you'll be able to look it up or you'll have a chart. But for example, PHE stands for the amino acid phenylalanine. SEA stands for the amino acid serine. PRO stands for the amino acid proline. Eventually you get kind of used to seeing these, you know what they are. All right, so the code is most often displayed in a table like this to enable you to find a specific codon quicker than having to scan through a whole long list. So for example, if I said CCA, you can use the code to look at the first, if I'm going to look up CCA. All right, so the first one is C. So the first base, I'm going to be looking at C, which means I'm going to be in this row. My second base is C, so I go up here, second base C. So I'm going this column, this row, and then here's my third one, A, CCA. So it enables me to go straight to CCA just by kind of triangulating between these sides, rather than having to read a whole long list until I get to CCA. And this tells me that CCA codon lines up with the amino acid proline. So this code tells you how to go from three-letter nucleotide codons to which amino acid is that telling the ribosome to add next. All right, so if the codon was ACC, this would be telling the ribosome to join in the amino acid threonine next. If it was AAA, it would be telling the ribosome to add the amino acid lysine in next. So ribosomes can understand these words because they have the tRNAs to act as like the bridge molecule. The tRNA anticodon will stick to the messenger RNA codon, and the tRNA will bring in the correct amino acid. Now, what that means is that for every single one of these codons, every single one of these 64 codons, you have a tRNA that matches it. So your body makes 64, actually it makes 61 different tRNAs. Why doesn't it make 64? Well, 3 of these are stops, and we don't have tRNAs that line up with the stops. So that's why there's a little difference. But your body makes multiple different types of tRNAs, and there's one to line up with every single one of these codons. So let's take the example here of CUG. And I'll draw it out here. If the codon was CUG, all right, and this is our messenger RNA, then my tRNA that would stick to it would have an anticodon of GAC, because these have to line up down here. And if we look CUG up in the genetic code, CUG is the amino acid leucine. So up here on the top would be LEU, the amino acid leucine. So this results in leucine being added in next to the growing protein chain, because the only tRNA that can attach to CUG is the one that has GAC on its butt. And tRNAs that have GAC on their butts will always have leucine stuck to their heads. Just how it goes. It's a pretty amazing system. It works incredibly well. But it relies on this specificity of having 61 different tRNAs, and that they all have the right amino acids stuck on their heads to make sure that whatever's down here is getting the right amino acid up there, so that when they're all lined up inside the ribosome, you get the amino acids lined up in the right order, joined together in the right order, and therefore the protein is in the correct order, because that's what's most important, is that protein's primary structure. Everything's put in the right order. Now, a couple of interesting things about the genetic code. It is absolutely amazing that every organism that we have so far looked at on the earth, it doesn't matter if you're a bacteria or a jellyfish or a fungus or a tree or a cactus or a human or a dog or a cat or, I don't know, an elephant. Every single living thing uses this same genetic code. We all speak the same genetic language. So AAC in a bacteria tells the bacteria's ribosome to put this amino acid in next. AAC in my messenger RNA tells my ribosomes to put this in next, this amino acid in next. So this is really strong evidence that all life on Earth shares a common ancestor. It's very unlikely that separately evolving pathways would have come up with the exact same words lined up with the exact same amino acids. It's basically impossible. So the odds are just way, way, way out there. So what we think happened is that some cell way back in the start of life came up with this way of lining everything up. It works well, and everything has used it ever since, and we've all diverged into all our different forms. Life has branched out after that, but we still use this same code. You can also, well, the benefit of that is that we can put genes from different animals into other animals or plants or bacteria, and the host organism can still read the instructions and make the protein that was meant to be from the original animal. So let me give you an example, and a really good example of this is in diabetes and insulin production. So if you go back maybe 20, 30 years ago, people that were diabetic who needed insulin, insulin was got for them from pig pancreases. So it was very expensive. It involved a lot of pigs. It was probably not there was more ethical issues with growing large mammals to create insulin from their pancreases. Plus, you are injecting porcine or pork insulin, pig insulin into you, and it's slightly different. The protein sequence is slightly different, almost identical, but a little different to human insulin. It works enough. Our cells can deal with it and respond to it, and it's good enough to treat diabetes for the people that couldn't make their own insulin, type 1 diabetes. But it was a little different, and people that had allergies to certain things maybe couldn't use it because it came from another animal, and there could be issues with that. So about, I don't know, maybe it's 30, 35 years ago now. It's definitely within my lifetime. Scientists were able to take the gene from humans for insulin, the sequence of letters, and put it into bacteria. And they could trick the bacteria into reading the instructions from the humans that they put into the bacteria, so the bacteria would make the human insulin protein. Now this is way better because bacteria are very cheap and easy to grow. You can grow bajillions of them in a vat rather than have to have a whole farm and pigs. They're small, they're cheap and easy to grow. They pump out insulin really, really quickly. There's no ethical issues with killing bajillions of bacteria like there are with killing bajillions of pigs. And the insulin that you get from them is 100% identical to human insulin because they're using the human insulin instructions, because the bacteria's ribosomes speak the exact same language as the human's ribosomes. And so the sequence in humans will translate to a certain order of amino acids. It's exactly the same if the bacteria read that instructions. Their ribosomes, their tRNAs will read all these words the same and make the same protein. And so that now is how we make all the insulin for type 1 and type 2 diabetics if they're on insulin. So if you see on an insulin vial recombinant or R in front of the insulin, that means that it's made from genetic engineering. That's what we're talking about here. And it was made by a bacteria, but it's exactly the same as human insulin because it's using the human insulin instructions, just having the bacteria do the job for us. And it wouldn't work if the bacteria didn't have the exact same language as we do. So that's the first thing that's kind of cool about it. In fact, I think that's the most cool thing about it. Anyway, the next thing just to notice is that the genetic code is what we call degenerate. And that means that there are multiple words that mean the same thing. So if we look at this amino acid here, proline, all these codons, CCU, CCC, CCA, CCG, they all tell the ribosome to incorporate the amino acid proline. And this is because there are 64 combinations of these letters, 64 codons, but there's only 20 amino acids. So you have extra codons for the amount of amino acids that you have. So some of them are doubled, tripled, even there's some of them that have 6, I believe. Like for example, arginine here. All these are arginine, and these two as well. Has 6. Serine too. These 4 and these 2. So there are multiple amino acids that have many different codons. There are a couple that only have one. AUG, our start codon, there's only one start codon. It's always AUG, and it's always the amino acid methionine. It always says put the amino acid methionine. You can have AUG in the middle of the sequence too. Like methionines can be in the middle of the protein chain, but they always are at the beginning. First amino acid is always methionine. And actually this one here, tryptophan, only has one codon, but methionine is the famous one. What that means though is that you can actually deal with some changes, some mutations will not have an impact on which amino acid is put into the growing chain. For example, it doesn't matter if it says CCU, CCC, CCA, or CCG, you're always going to get the same amino acid. And what really matters in proteins is that the amino acids are in the right order. So as long as you got a proline there, it doesn't matter if you use this codon to put it there or that codon to put it there. So this degeneracy, this third base, it could be either any of the four, provides some protection against accidental changes that could change the whole amino acid. You don't want to change the order of the amino acids, that's bad. And most of the time, changing one of the letters will cause a problem. But if you accidentally change the last letter of a threesome, you might be okay. Not all the time, but you might. And that's what we mean by when we say the genetic code is degenerate. We'll say there's often more than one codon for each amino acid. All right, so that's a little bit about the genetic code.